Hello, still on our class in germination. Germination is a process by which a seed begins to grow and develop into a seedling. Let me quickly refresh our mind on the terms that are associated with germination. We hear words like plumule, cotyledon, hypocotyl, epicotyl, radicle, seed coat, or you call it the tester, etc. These are the terms that you likely hear, including the endosperm, the coleoptile. Do not forget that germination is divided into two: the apigeal germination and the hypogeal germination. On either of the type of germination, the same steps are involved in germination. Number one step is seed activation. It simply means that seed need to be activated. Once a seed is planted in the soil, it needs to be activated by light or water or any other environmental factor. Once it's been activated, the seed absorbs water or moisture, as the case may be. Once it absorbs water, the seed coats will now soften and swell up. Once that happens, the seed coat will crack. Once this happens, it will now allow the radicle to emerge. The radicle is what forms the primary root, which we also know as the taproot. Then hypocotyl emerges. This hypocotyl is what keep pushing up the cotyledon above the soil level, which on appearance above the soil level straightens. The cotyledon itself is a seed leaf. Another name for the cotyledon is a seed leaf. It also emerges from the seed and they are carried above the soil level in apigeal germination. And upon appearing above the soil level, begins photosynthesis. Now the seedling begins to develop further and its root system begins to develop and becomes a matured plant. So with all these that I've mentioned, it will interest you to know that there are some factors that affect germination, or you can say conditions required for germination to take place. Number one is water. Water is very essential for germination. Second one is temperature. Temperature here, it has to be the optimal temperature. That word optimal temperature simply means the required temperature. And this temperature varies with seeds. Different seed has the optimal temperature required in order to germinate. But ordinarily, most seed germinate from 65 Fahrenheit and 85 Fahrenheit. Another element is the soil quality. Soil quality. You can agree with me that it's not every soil that is good for agriculture. You cannot successfully plant on a sandy soil and expect miracles. Let's not forget the quality of the seed. Quality of seed, or you can say the viability of the seed. A seed that has been eaten up by insects or birds can no longer germinate. Let me draw your mind to what I mean. For example, your beans. If you look at a beans and you can see that there are holes inside the beans, look closely, notice that the whole contents have been eaten up by bean weevils. Okay, so all this now will help us to draw a conclusion on the tips for a successful germination. Number one, use fresh seeds, as old seeds may have some kind of lower germination rates. Due to what I've explained earlier, it could be that the content of the seed has been eaten up. Then, optimal conditions. It's important to research and find out the condition that are specific for each seed type. Very, very important. I remember giving students a project to cultivate a bean seedling and a corn, and after some days they came back and said that it refused to germinate. It's possible. Some may have done it and left it under the sun, directly under the sun rather, and everything, including the water and the seed, may dry up because the condition which it found himself is not suitable. So it's important that you monitor the temperature in order to promote germination, and most often the temperature has to be consistent. So as a young developing seed, it's important to do what? Follow all the right steps in order to ensure successful germination. So there's another term that I skipped to mention. It's called the coleptile. Coleptile is associated with monocotyledons. They are like protective sheets for the plumule. So if you observe when a monocot germinates, let me use maize plant as an example, you notice something like a long green sheet growing up. So that long sheet is the first to emerge in a monocotyledonous plant in order to protect the young leaf inside. So as soon as the condition becomes favorable or the young plumule is able to handle the harsh environment, the coleptile gives way and the plumule emerges completely. So coleptile is a protective sheet covering the emerging shoot in monocot seed. Coleptile, they are what? Protective sheets that covers emerging shoots in monocot seed. Also, the endosperm and the cotyledons, we need to understand that it is not every seed that has endosperm. Dicotyledonous seed are associated with the word cotyledons, while monocotyledonous seed are associated with the word endosperm. So it's not every dicot. In fact, most dicots, they don't have endosperm. Endosperm are mostly for monocotyledonous seed. This is because as monocots, their cotyledons are not carried above the soil level, and as such, the required nutrients for their growth are embedded in the endosperm. So this is what they continually feed on until the shoot, which is being protected by the coleptile, emerges completely and starts photosynthesis. So for the dicots, they don't have the endosperm. Rather, what they use for photosynthesis, the cotyledons, that's what they use, which is carried above the soil, and upon reaction with the air turns green and starts providing food until the first foliage leaf appears. So note the difference between the monocots and the dicots. In case you have forgotten what the monocots and dicots means, monocots are those seeds that have one seed leaf, Why the diacots are those seeds that has two seed leaves. I've given examples of the monocots, which includes the maize, the millet, etc., and the diacots, which includes the castor oil seed, the bean seed, that's from the cowpea family, the groundnuts, etc. So, as a way of rounding off, germination is divided into two, remember? Apigeal and hypogeal germination. In apigeal, the cotyledons are carried above the soil level, while in hypogeal, the cotyledons remains below the soil level. You can say they remain underground. So in our next class, we'll take up that of hypogeal germination. We'll explain it the same way we have gone through that of apigeal germination. Thank you.